Today we're going to do a tutorial on southern blotting. My name is João, and what we're going to talk about, or what I need to tell you about this technique here, is that it's used to detect a specific DNA fragment in a mixture of other DNA fragments. And we are going to discuss this technique in a lot more detail on the following slides. Now, the first thing that I thought when I looked at this name here, southern blotting, I thought this was discovered or invented somewhere in the southern hemisphere, somewhere in a southern location. I was completely wrong, and I felt like a fool. No, it was a British biologist of the name Edwin Southern that came up with this amazing technique. Now, blotting, that's a trickier one. Blotting means transfer. So when you decide to transfer a DNA, an RNA, or protein from one carrier onto another. Now, on southern blotting, the blotting process is done, or the transferring process is going to be done, or we're going to transfer DNA molecules from a gel to a membrane. And the membranes can either be nylon or nitrocellulose membranes. We're going to see that in a little bit more detail later on in this tutorial. Now, the three last points that I have here is to illustrate something that will give you a better or a clearer view of what southern blotting is or does. Well, southern blotting is basically a mixture or a combination of different techniques used in genetics, let's say, to find a specific DNA fragment. And these techniques are gel electrophoresis, blotting or transferring as we saw here, and probing, when you use a DNA probe with a radioactive probe, let's say, or radioactive DNA probe, better said, and to detect the specific DNA fragment that you wish to. Now, if you remember well from the previous tutorials, this means that southern blotting is basically a combination of two other major techniques, and these are restriction mapping, if you remember well, and also molecular hybridization. And we're going to see why on the following slides. Now let's talk about the steps of southern blotting. First thing that I should mention before we go into detail is that there are 7 defined steps for southern blotting. Now, the first one is what you have here, this illustration that I'm going to use in a little bit more detail to explain the first step, which is cleavage of DNA by restriction endonucleases. So this is the very first step. And you have a DNA molecule here. And what you're going to do is use restriction endonucleases, these specific enzymes that are able to cleave DNA at specific sites. They recognize specific palindromic sites and then cleave and constitute these fragments that you see here. So this is the very first step that you do in southern blotting. So on the first step, what we saw was a DNA molecule being cleaved by a restriction endonuclease, or by restriction endonucleases. What we are left is with these fragments that you see here. So the second step will logically be separation of this mixture of these fragments here by agarose gel electrophoresis. And what happens is if we take the gel that you see here and take a mixture containing these fragments and say start here, after we start running the gel by using a current, meaning that we're going to have two electrodes, one negative and one positive, and due to the fact that the DNA backbone is negative due to the phosphate groups, you're going to see that these fragments are going to migrate towards the positive electrode. Now what you should see is something along these lines. Well, in actuality, you will not see it. So the DNA fragments are invisible to the naked eye, and that's why you need to proceed with the next few steps in order to visualize the separation of these DNA fragments right here. Another important thing to notice is that these DNA fragments are separated by size. So the largest are usually left behind, while the smallest fragments are able to travel faster. Second step, what we did was simply separated the DNA fragments based on their sizes, but we're still not able to visualize them on the naked eye and detect the specific DNA fragment out of those three that we saw. We cannot do that. For that reason, we have to proceed, and the third step then is going to be blotting. So this is our third step, transferring those DNA fragments onto a nylon or nitrocellulose membrane. And I have here what it's supposed to be a basic setting for blotting. And what you see here, and I'm going to try to locate the gel. So this is the gel containing the DNA fragments. And this pink structure that you see here, this is representing the filter or the membrane. So you want to transfer these DNA fragments from the gel to the filter. You have here then a sponge and a salt solution or a buffer, and on top you're going to have a stack of paper towels. So easy setting to have in a lab. Now what's going to happen here, there are two things that are going to happen, and one I need to tell you that it's going to be also the fourth step, and this is DNA denaturation. So there's going to be DNA denaturation due to the alkaline treatment that can happen as well here during this transferring or sometime before or after, depending on the technique or the protocol that you're following. But what's going to happen is that you have to, after the blotting or after transferring or before, you have to denature the DNA. Go from a double-stranded to a single-stranded DNA form. That way you're going to be able on the next step to bind the DNA probe for detection. Now what I want to say, and back to the blotting, because this is just to mention that it can happen either before or after depending on the protocol. But on the blotting, what's going to happen is then the DNA is going to be transferred to the filter by a passage. So it's going to be transferred this way by passage of the salt solution through the gel. So the salt solution is going to be absorbed in this sponge here, and then to the point of pushing, let's say, the DNA fragments, and then they're going to end up being transferred to this filter right here or to this membrane. So this is the blotting process of southern blotting. So after blotting, I'm left with a membrane containing the DNA fragments, more or less on the same position. And as I mentioned before, these DNA fragments right now, and due to the fourth step, are now found in the single-stranded form. So I denatured the DNA fragments with an alkaline treatment. Why did I do that? Because the next step, the fifth step, is hybridization with a label probe, a radioactive DNA probe. And why do I do that? Because, as I mentioned before, southern blotting is a detection technique. So I want to detect a specific DNA fragment. So what I do now, and if you remember well from the tutorial on molecular hybridization, I explained there that we can use a radioactive DNA probe, which is an oligonucleotide containing a radioactive labeling. That way we can visualize, detect where this probe is, and this sequence here is known. So this is a known sequence. And what's going to do when we expose this membrane containing these DNA fragments, this sequence, this known sequence, is going to be able to find the complementary sequence either on this band, on this, and on this one. And depending on which one it's going to bind to, then we're going to be able to visualize. We're going to be able to detect, in this case, since I use the green for the radioactive label, so we're going to be able to say if the complementary sequence is found on the second band, then I should find it here. And this is how you visualize. This is molecular hybridization. But we're going to get to this point. Right now I want to discuss the sixth step, and the sixth step is washing of the unhybridized probes. So after we hybridize them, meaning that we're going to expose this membrane containing the fragments and allow this probe to bind to the complementary sequence, then we wash those probes that haven't been or haven't found a complementary sequence. So on the seventh and last step of southern blotting, what we do is we have a look or visualize where that specific DNA fragment is. And we do it through auto radiography, if you remember well from our molecular hybridization tutorial. And what I have here for you is a illustration. Again, I'm not an amazing artist, but this is to illustrate the membrane here before. So this is before we look at it with autoradiography. So we know that the probe has found in one of these bands has found complementary sequence bound to it, and now we have to be able to observe under autoradiography, and we're going to be able to see the radioactive probe and find where the DNA fragment that we wanted to find, that specific DNA fragment that we want to find is. So this, my friends, is the magic of southern blotting. This is how you are able to detect a specific DNA fragment using this technique.